Good morning, everybody. Today we have this kind of flash seminar. Maybe the date was not the best because it hits the pre-day of the national holiday of Estonia. So probably many people are not here because of this reason. The idea for this talk was inspired by the finished IMC12 symposium, which was important in this respect that many nomenclature and taxonomy related issues of fungi were discussed. So I will shed some light about what was going on. So share my screen. I had also a talk about eDNA-based taxonomic applications in this symposium. So to get into business, the code of nomenclature for plants, fungi, and algae sets the standards how fungi should be described and principally what are the rules. So it says that species can be described based on either a description or a diagnosis. Diagnosis is a type of description as well, which is intended to tell apart the new species and previously described species. The holy grail in taxonomy is a holotype. And the code says that it can be a vouchered specimen, killed culture, even a figure, or also a DNA sample. So it can be quite broad. And further descriptions of genus, family, order, and class must rely on the type species and accordingly the type genus, type family, etc. Interestingly, the description of a new phylum is not regulated by the code. A few years ago already, a few proposals for improving the nomenclature were prepared for this Congress. So first, sequences should be allowed to act as holotypes. Also in these cases, a taxonomically valid marker or at least ITS region should be used. However, about 70% of the participants voted against this proposal. And in the final voting, the proposal was withdrawn. Second proposal was to allow genomes to act as holotypes. Unfortunately, this proposal was ill-prepared and it didn't specify very many important things. So it was principally with very limited restriction. So the most concerning for me was that the genome might be set as holotypes without any ITS or other marker sequenced, and no description or diagnosis based on this genome was required. Initially, about 60% of people voted for this proposal, but after more discussions, most of the people decided that it is premature, and they suggested that a specific committee for the genome-based description should be put forward. In parallel, since some time ago, we have also started our work on eDNA-based classification. Part of this is reflected in the UNITE database, which uses species hypothesis and also taxonomic hypothesis for marking higher level fungal taxa. Unfortunately, this taxonomic hypothesis concept has not become popular. In this work, we have also tried to describe new fungal lineages, and more recently we have found that it might be important to also describe the species to anchor these lineages. So our approach is the following. So first, we should focus on the OTUs from eDNA samples that cannot be identified to some higher taxonomic level. Also, we should only focus on those groups that are expected to be unculturable, or that cultures just don't pop up here and there by chance and ruin this new system. Then, based on a phylogenetic analysis, we nominate a new species, potential new species in the crown group of this lineage, and this new species must have multiple independent records. So it means records using different primers from different samples, preferably also from different projects, and also from different teams, including previous data published in GenBank. Then we would select representative sequences and its eDNA sample to serve as a holotype. Based on the delimited species, we would prepare a diagnosis based on the sequences and focus on a short mini barcode, some 20 to 30 bases in length. So this should be very unique among all fungi and easily and very rapidly recognizable by software, and also understandable to a naked eye. It's also important to estimate interspecific variability to understand how the species in this particular new genus could be bordered or delimited. So this will come from sequence data and its variation and comparison to the closest related species. Important step is also to consider a name and epithet for both genus and the species itself. Our approach has been focusing on collectors of the samples or the people, researchers, and localities where these type species or type sequences or eDNA has been found. And then, based on the described species and genus, we would describe higher ranking taxa using the least inclusive lineages approach based on the phylogenetic analysis. So to make the first exercise for this, we prepared such kind of classification for vascular mycorrhizal fungi that includes Glomeramycota, but also Endogonomycetes of Mucoromycota. We relied mostly on the sequences accumulated in the eukaryote database. So it includes a large proportion of very long reads produced using PacBio third generation platform. On the left, you can see the tree for Glomeramycota, and here we found one new group close to Enterophosphora, which we named as pseudo-Enterophosphora. And we found that it is sufficiently divergent from the genus Enterophosphora. So based on the species Pseudo-Enterophosphora gesseensis related to its type locality, we also described a new family, Pseudo-Enterophosphoraceae. And regarding those miniature barcodes, we defined one for the ITS region. You see the top 4 sequences represent this species. And we defined it also for the LSU part. Again, the top 4 sequences. So in this case, they are all in agreement and don't have any mismatch or difference in this region. A more complicated story is related to the endogonomycetes based on using specific primers and PacBio sequencing. We were surprised to see that this group is extremely broad and heterogeneous. We found evidence that there are 16 order level groups. And first we described the one species for each of these groups, followed by describing corresponding families and orders. For the rest that couldn't be described, we produced an alphanumerical naming system, which you can see on the tree. So these serve for initial communication, and these might be the backbones of new species, genera, and families to be described in the future. Also on the tree, you can see that there are a few other taxa that are not assigned to any of the described orders. So this might be also a good seed for future work. And in the right figure, you can see again the diagnosis for a new species along Dua Diana and comparison to other more than 30 species of the same new genus. Actually, we started this kind of work and coding of new lineages 6 years ago. And then there we saw that there are at least 3 phylum level groups that cannot be accommodated to any known phylum. So these are basal clone group 2, basal clone group 1, and clade GS01. Again, using the accumulating data from eukaryote, we elaborated on this previous study, And using long reads, we found that there are actually many more groups at the level of class and phylum, not just described in the fungal kingdom. For example, there is one small group, basal clone group 2, which seems to be consistently occurring next to the kingdom fungi. Okay, so there are several new groups close to aphelids and also new groups close to chytrids. For example, a sister group to monoblephs, which was recently also characterized by single cell sequencing by Keisuke Seto and colleagues. There might be one new phylum within the chytrids and several new classes related to Neogallimasticum mycetes and within Chytridium mycota sensu stricto. The large group GSO1, based on new analysis, seems to be more close related to Plastocladio-mycetes and some Chytridia mycota. Also within the Zoopago mycota sensulato, there seem to be quite many new phylum and class level groups. The Olpidium mycota that we know based on a few terrestrial plant pathogens seems to be also a huge and very diverse group, potentially also including many algal pathogens and many new classes and especially new orders. Close to Calcaris borealis, there is another new order level group undescribed, and at the base of Mortiorellales, there are 3 order and class level new groups. And finally, next to Glomeramycota, there is yet another phylum level group, which is interestingly also mostly found from root samples. And in this figure, I don't display minor groups within the Phasidio and Ascomycota, but individual trees suggest that there are something between 20 and 50 groups at the level of order and around 10 groups at the level of class that are not described. Elaborating further on these new fungal lineages, we first took this group next to Glomeramycota under attention, and we produced the phylogenetic tree, nominated the crown group and potential new species, which we putatively call Slava Ukraini Kharkivense, and we nominated again the mini barcode for both ITS and LSU. So since I presented this work in quite early days of this one week conference, I managed to get a lot of feedback to some especially old guys in the IMC12 said it's bad science, stop it, it's non-reproducible. Some people argued that mixed eDNA samples should not act as holotypes. Higher ranking taxa should not be described based on the least inclusive clades. Many questions about naming priorities. What if a new name comes based on a cultured and redescribed specimen? And of course, also there is a risk of describing unsequenced valid species. So at least partly, of course, I agree with all these constructive criticisms, but I believe that the thing we are running at the moment, it actually is a good way forward to make also other people think and go forward with the nomenclature, taxonomy, and fungal classification. So to get also some anonymous feedback, we distributed a questionnaire about the related thoughts, and we will analyze it soon and also evaluate how researchers with different age and position think about these problems. So to conclude about this, eDNA related sequences can make a huge contribution to the tree of life, especially when they originate from long reads and single cell analysis. Principally, the same might apply for taxonomy and classification, but certainly we need to set the rules. eDNA can also indicate the best metabarcoding markers. So, for example, in the study of avascular mycorrhizal fungi, we saw that LSU works better than ITS, and together they work much better, both in glomerulomycota and endogonomyces seeds, both from the perspective of discriminating among species and phylogenetic resolution as well. In particular, the benefits of long-read sequences again include greater taxonomic and phylogenetic resolution and building of real phylogenies. Long reads can also reveal primer biases and any reasons why ITS cannot be amplified in certain taxa. For example, in many groups, the ITS region can be super long, or the common primer sites next to the ITS region are heavily mutated. Unfortunately, long reads are more prone to accumulation of various errors and also construction of chimeric sequences. So these need extra care. So with this, I stop sharing the presentation. And of course, I'm happy to hear any feedback from you and discussion what we might do further. And of course, if any one of you is interested in describing, for example, new taxa in Basidiomycota and Ascomycota using this kind of approach, we can discuss it later. So I see hand of Irina, please.
Hello, Leho. I was also at IMC and I heard emotional discussions on this point. Yeah, and about this work that you presented, of course, first of all, Slava Ukraini, the genus and the species, we all were very flattered, and I think it is fantastic, and everybody approved it, at least from our side. And secondly, I agree that the dark diversity should be described in some way because, well, it exists and we cannot just ignore it. The question is how it should be described. and this work, at least the trees presented in this work, seem to me problematic because they support the methods, the supports of the trees are quite not very reliable, because this output that the ultra-fast bootstrap that was used is quite unreliable method, and it doesn't really show much, especially if one region was used. So basically, I mean, it's a good way to provoke a discussion, but not the best way to describe taxa. Yeah. And what do you think about the alternative that was suggested by IMC12 that would be analogous to candidatus in bacteria? It was called nomsec or something like that. So the code would be this names based on eDNA would be not formally described, but something like provisional, like in bacteria.
Yes, well, I've been thinking of these possibilities too, but the main problem is that if you go with candidatus and nomsec, it would mean that principle you generate an alternative taxonomic system, and this has partly happened in bacteria, where people who were frustrated that they cannot describe species based on sequences, they generated their own system, sec code. So now in bacteria there are like two parallel systems. Of course, they try to see that they are not heavily mismatching to each other, but still it's a weird situation, and I would like to avoid this at all cost. The solution might be that if we describe the species on top of everything, we can also add this nomsec, so that could be some kind of compromise.
I don't really understand why there is the need to use the code that is really the purpose of the code is really hooked on the specimen, not on the sequence. And I'm afraid that the amount of a new taxa that are not really stable will be, if the formal naming is allowed, that will be fixed forever, and it will just create a lot of huge amount of work to the further generations. Because if the formal description is allowed, then we will be forced to consider all the names formally and spend a lot of time disentangling it. So that's, I think, it's my consideration, and many of participants of the IMC12 also like thought in this line.
Yes, of course, here I completely agree, but it also depends on the situation. Well, obviously, nobody would go and describe a new species of Aspergillus or Penicillium or even, let's say, Russula based on this kind of eDNA approach. But it should be somehow limited to those completely undescribed groups for which we don't have any culture, we don't have any specimens, we don't even have anything sequenced from something that could be served as a specimen. So that's this kind of different story, and it's probably even in the next 10 or 20 years we can't move forward fast enough. When we first published the new fungal lineages, it was 7 years ago. Since then, out of those 30 new lineages, only 2 have been described officially, and in both cases their work was partly inspired by our work of nominating those groups. So then they rediscovered that indeed it can be a taxonomically very valuable specimen, and let's describe it quickly rather than in 10 years.
Well, I agree that this should be very clearly separated into the taxa that could be formally described as a specimen and those that cannot. But still, in this proposal, it was not clear. This proposal was full of holes that allowed to describe whatever, basically, based on DNA, even if it's culturable, even if it's can be collected. So that's why I think this proposal were like rebutted quite heavily, because they have not well prepared. Okay. Thank you.
Okay, thank you. Do we have any other questions or comments?
I have a question.
Yes, please.
How do you handle the interspecific variation in the sequence with the error accumulation from the platform?
Yeah, that's certainly a critical question here. We try to use principally all unique sequences for describing the species which were generated in this platform. So principally, if we do the sequence generation by ourselves, we can do it. So not rely on a single representative, but take into account all of the sequences produced. Of course, those very obvious sequencing errors we can misconsider and nominate or assign these sequences as low quality or erroneous and throw them away. But in principle, yes, we should take into account all these sequences that are produced and find this best region for generating this mini barcode, which is either as homogeneous as possible or that presents some critical polymorphism that could be also characteristic. And when it comes, of course, to the full-length sequence, we can just, let's say, take one of the most divergent sequences and principally count the number of mismatches or mutations in other sequences, and based on this specify the interspecific variation in this particular region.
Okay.
All right, so if I don't see any other questions, then we can also happily finish this flash seminar. It was not intended to be a long discussion. Of course, it generates a lot of thoughts and many things cannot be obviously set out or discussed in this very short time, but I'm happy to discuss with any of you separately about these questions. Okay, Muhammad still has a hand, please.
Good morning, everyone. Professor, actually, um, I think it's my like not a question maybe or just my thoughts about that about eDNA. Like the main thing, like we are facing the limitation challenges, or like you said, there is a lot of like in a conference they said it's a bad science, you have to stop it or you have to change it, something like that. Is it on the base of that limitation? Because what I know about like Glumicota or this phylum, like there is a lot of like very less data is available on that particular topic due to the root zone, because the study on this topic is very limited and database is very limited in that term. Is it the whole scenario or the whole feedback which is a quite uh, not so much positive feedbacks? Is it due to the limitation, or what type of questions are rise on this conference? Like what type of question they ask, like this is the loophole or this is the things in that? Because I think there is already very less data available on that topic already.
Yes, there is quite little data available, but it really depends on how you search for it. If you dive into, then you can find a lot, because there are also all those short read archives and the metagenomic bins and everything that you potentially might approach. But yes, regarding those feedbacks, well, there was really various concerns. There was also a lot of support, especially from the younger generation and from people who were also handling those dark taxa lineages. But I didn't bring these here because it's more important to learn from constructive criticism. So these were the main points, and of course, in the future we need to handle these as good as we can, especially when we are going forward with this global fungal tree of life. Well, okay then. Thanks for attending. Discuss to you later if you have questions, and have a nice day and happy nice holidays tomorrow. See you.